Today, I'm going to talk about some of the work that I've been doing in my lab, focused on population genetics. But more broadly, my interests center around making sense of genomic data using techniques from statistics and machine learning. And we have a few different directions that we work on. So one set of directions is focused on using machine learning to analyze clinical data and to improve treatment outcomes. Another set of directions that we work on is focused on the genetic architecture of complex traits. So this was what I discussed yesterday. But in today's talk, I'll focus on evolutionary questions that we are aiming to address by analyzing genomic data. So I'm going to talk about primarily admixture or introgression from archaic populations, so populations that are quite diverged from modern human populations, and what we've learned about these events and what kind of impact these events have had on biology. And so this talk is structured in 4 parts, which will discuss different aspects of this question. And part of my goal here is to tell you a little bit about the kind of findings and to summarize what we've learned, but also to give you some idea of the statistical and computational techniques that are underpinning these questions, just to give you an idea of what is interesting at the intersection of biology and computation. So we're going to start with looking at the history of modern and archaic humans. So we're interested in understanding modern humans and how they relate to Neanderthals. So based on analyzing genetic sequences and from the fossil record, the main picture that arises about the dispersal of modern humans is one where modern humans originate in Africa around 200,000 years ago. And then there was a dispersal out of Africa between 50 to 100,000 years ago that gave rise to the ancestors of present-day humans outside of Africa. Now, based on the fossil record, we know that there was another species, another population that existed, the Neanderthals. They appear in the fossil record around 300,000 years ago, and they disappear around 30,000 years ago. And the fossil record of Neanderthals encompasses large parts of Europe and Central Asia. So there was overlap between these two populations, both in space and time. And so the question that arises is, as modern humans were spreading to different parts outside of Africa, what was the interaction between these populations? Was there interbreeding? And what was the outcome of this interbreeding? So there have been many attempts to answer this question, but one of the most definitive ways of doing this was by looking at genome sequences of Neanderthals. So the draft genome sequence of the Neanderthal was published in this paper in 2010. And one of the key findings that came out of analyzing these genome sequences is if you take the Neanderthal genome sequence and compare it to the genome sequence of an African compared to a non-African individual, Neanderthals are genetically more similar to non-Africans than to Africans. So this pattern, which is observed no matter which African and which non-African populations we compare against, suggests that interbreeding could have been a possibility in this dispersal of modern humans out of Africa. So as we were starting to think about this question of interbreeding, one question that came up was, this was not the only possibility. So although non-Africans are genetically closer to Neanderthals than Africans, and this hinted at the possibility of admixture, there are other possibilities that could also explain this observation. So this was the possibility of complicated population structure. So in this model, populations in Africa have very deep structure, where you can imagine 3 different populations that are not entirely panmictic, they're exchanging genes. And one of these populations moves out of Africa into Europe, becomes the ancestors of Neanderthals as we know them. And then you have these 2 other populations, one of which was closer to the ancestors of Neanderthals, and that becomes the ancestors of present-day non-Africans. So these 2 models, they can both explain this genetic similarity, but they do make one difference in terms of their prediction. And that was the date when Neanderthals and present-day humans last exchanged genes. So in this ancient structure model, the date when the last exchange of genes happened between modern humans and Neanderthals goes back to when modern humans appeared in the fossil record. So this would be around 200,000 years ago. On the other hand, in the admixture model, the date of last exchange must have happened after the divergence of non-Africans from Africans. So this must have happened within the last 100,000 years. So one way to differentiate these hypotheses is to try to estimate the date when Neanderthals and modern humans last interbred. So to get at this, we can use the following property of admixture. We use the property that as genomes are transmitted from parents to offspring, there is this process of recombination, which breaks up the genomes that get transmitted from one generation to the other. So in this cartoon, if you look at the first generation of the offspring of a modern human and archaic, you will have entire chromosomes that are inherited from one or the other population. But as time progresses, because of recombination, the chromosomes that the segments that get inherited break down. And essentially, the longer the time since admixture, the shorter the length scales of these segments. So the length scales of these introgressed archaic segments can be used as a molecular clock to be able to date the last exchange of genes. So we applied this strategy, and instead of measuring the length scales of these segments, which turns out to be technically challenging, we compute a statistic which is related to the correlation coefficient between pairs of adjacent genetic markers. So this is related to linkage disequilibrium, or LD. And we asked how this correlation decays with genetic distance. And it turns out, using some theory, we can show that this correlation has an exponential decay with genetic distance, and the rate of decay gives us the time of admixture. So by measuring this decay constant, we estimated that the date of interbreeding can be traced around 50,000 years ago with fairly large confidence intervals. And this large confidence interval comes about because of different sources of error that we have to integrate into in this analysis. Errors in these genetic maps that we're using to measure distance, as well as our lack of certainty about generation times when populations. So with this, we can conclude that interbreeding between humans and Neanderthals happened fairly recently, certainly after the out of Africa migration. We can also estimate what proportion of genetic ancestry of these individuals traces itself to Neanderthal. And what we can estimate is about 2% of the ancestry of present-day non-Africans traces itself to this introgression. So with this, we get some idea of the global dynamics and the global picture of Neanderthal introgression into modern humans. At around the same time, there was this question that, given that these Neanderthal segments were quite different genetically from the modern human genomes that they were introgressing into, what impact might they have on human biology? So one of the first hints of this came about from another strand of work, which was a genome-wide association study. So this was a study that was trying to identify genetic risk factors, in this case in type 2 diabetes, in a population of Mexican Americans. And this analysis identified a novel genetic risk factor found in this gene, SLC16A11. And when we look at the geographical distribution of this variant, we find this interesting pattern where the risk variant is essentially absent in Africa. It's present at low frequencies in Europe and parts of East Asia, and at appreciable frequencies in the Americas. So this is the kind of geographical pattern of allele frequencies that would be consistent with this haplotype being introduced, this variant being introduced by introgression. And indeed, when we compare this variant, which lies on a haplotype, so there's a combination of this variants associated with type 2 diabetes, we find that it essentially matches the genome sequence of the Neanderthal that was sequenced at that point of time from the Altai Mountains. So this was one of the first examples of a specific genetic risk variant inherited from Neanderthals tied to a increasing disease risk. There have been other notable examples of introgression contributing to biology of specific loci. So there were early studies that looked at STAT2, which is the locus that is important for immune-related function at the HLA loci. And all of these documented fairly significant contributions from archaic introgression at these levels. But what was missing from these initial assessments was going from individual loci of genes to a more genome-wide assessment, trying to understand across the genome what was the impact of introgression. To get at this, we need to build maps of Neanderthal DNA. In other words, we want to be able to locate regions of an individual's genome which have tracing their ancestry to Neanderthal introgression. So to build these maps of Neanderthal ancestry, we rely on statistical techniques, which essentially compare a test genome, a genome from an individual of non-African ancestry, to an archaic genome, say a Neanderthal genome, and to a modern human genome that lacks any archaic ancestry, for example, African genomes. And by comparing the patterns of variation in the test genome to the archaic and the modern human genomes, our goal is to be able to label these segments of DNA as whether they are inherited from Neanderthal introgression or not. So in building these models, these statistical models to predict Neanderthal local ancestry, we use several features from data. I won't talk about all of them, but here's an example of the kinds of features that we use. So one class of features look at patterns of genetic variation at individual SNPs. So here is one such SNP where there is a mutation, a derived allele, that is present in the Neanderthal genome, that is also present in a non-African genome. And this allele is missing in all the African. So seeing this pattern of genetic variation makes it more likely that this segment in the non-African population is Neanderthal. Now, there are other types of information that we can potentially use. So we can go beyond individual SNPs to looking at patterns and combinations of SNPs, which we call haplotypes. So we can ask, are there haplotypes, combinations of SNPs, which render the non-African segment closer to the Neanderthal than to the African genome? And finally, the time of admixture that we estimated to be around 50,000 years gives us an expectation on the sizes or the lengths of these segments. So we have these different sources of information and we put it into a statistical model that combines these different sources of information to label or predict Neanderthal local ancestry. So the details are not too important. This is for those of you who are familiar with hidden Markov models. The model that we use is a similar version called a conditional random field that allows us to combine potentially correlated features to make these inferences. So applying this method, we are able to build these maps of Neanderthal ancestry at an individual level. So here is an application of this method to data from the 1000 Genomes project. What you see here is one single chromosome, chromosome 9, and you see the probability of Neanderthal ancestry as estimated by this model for a European individual, an East Asian individual, and as a negative control, we also applied it to a sub-Saharan African genome. And what we see is that there are several places across the genome where the model confidently infers Neanderthal ancestry for the European and East Asian genome. Relatively few such places in the African. So this is consistent with what you would expect based on our model of Neanderthal human introgression. This is also seen when we look at genome-wide proportions that we estimate from this model. So if we count up what fraction of the genome is estimated to be Neanderthal in ancestry, we find that there is substantial contributions that we expect from European and East Asian and substantially lower in African genomes. We also did some additional analyses to quantify things like the false discovery rate, the recall or the power of this model. And we can estimate that at false discovery rates of around 20%, we have anywhere between 60 to 80% power to be able to identify these segments. So now we can go from these individual maps of Neanderthal ancestry to aggregating them within a population to build population level maps. Here we are building population level maps in European and East Asian populations with the 1000 Genomes project. And what you see here is as we move across the genome, we are counting up the frequency of Neanderthal ancestry in each of these populations. And what we see here is that there is considerable variation in the frequency of Neanderthal ancestry as we move across the genome in these two populations. So on the one hand, there are places where there is increased frequencies of Neanderthal ancestry, what we call peaks of Neanderthal ancestry. And then there are regions in the genome which are essentially devoid of Neanderthal ancestry. We term these deserts of Neanderthal ancestry. So here's an example of one such peak. So this is a region where today about 60% of European individuals carry Neanderthal ancestry. So this has to be compared to the 2% Neanderthal ancestry that was introduced into modern humans about 50,000. So this peak of Neanderthal ancestry overlaps with gene BNC2. And this is a gene which has been shown to be associated with skin color, pigmentation, and hair color. So we did some additional analyses to ask whether these peaks of Neanderthal ancestry could have risen into frequency neutrally, or whether there is selection involved in increasing the frequencies of these regions. And it turns out for these peaks of Neanderthal ancestry, it's fairly clear that they must have risen to frequency, these high frequencies, by positive selection. We can also attempt to figure out what might be the biological processes that are associated with regions of elevated Neanderthal ancestry. So we looked at gene ontology analyses of these regions, and we find that they tend to be enriched for skin and hair color related functions, skeletal biology. Now we turn our attention to the deserts, the regions of the genome which are essentially depleted in Neanderthal. Some of these regions are actually quite big. So there are these multi-megabase windows which are devoid of Neanderthal. It's hard to figure out why and what's the mechanism that's driving these deserts of Neanderthal ancestry. But one such desert, which is quite prominent, is a desert on chromosome 7 that overlaps something called FOXP2 gene. Again, it's a large region. There are several genes there, but what makes this interesting is this is a gene that has been shown in prior studies to be important for speech and language-related processing. So a conjecture at this point is these deserts of Neanderthal ancestry are regions which are resistant to introgression, possibly because they have functions or they encode for functions that are important for modern human behavior. Another way of looking at this is a quantitative approach where we can divide up the genome based on a quantity which is called B statistic. So the B statistic is a measure of background selection, so a measure of functional constraint across different regions of the genome. And we can ask what happens to the Neanderthal ancestry as we move across values of the B statistic. So as we move from regions of high B statistic, which are neutrally evolving, to regions of low B statistic, which are under selective constraint, we find that the proportion of Neanderthal ancestry is decreasing both in Europeans and in East Asians. So this is consistent with purifying selection acting on introgressed ancestry. So the question is, what might be driving these regions where there is depletion? So there's been some work coming out of Graham Cook's lab and Nielsen's lab, which shows that this purifying selection against Neanderthal ancestry is driven by the fact that when we look at Neanderthal population sizes, historically, they were quite small compared to most modern human populations that we're aware of. And as a result, there's been an accumulation of deleterious variants in these populations, which then enter into the modern human population and get removed by selection. So this is one explanation for these deserts of Neanderthal ancestry that we observe. Another possibility is this notion of incompatibilities, or what we call male hybrid sterility. So the idea is that there are genetic variants that accumulate on the Neanderthal and the modern human lineages, which are neutral in isolation. But when they come back together into a single population, then these combinations of genetic variants are deleted. So this was a hypothesis that we tested by looking at the Neanderthal ancestry and how it is distributed on the X chromosome versus the autosomes. So the hybrid sterility model makes the prediction that genes which are involved in hybrid sterility tend to be concentrated on the X chromosome versus the autosome. So when we look at the Neanderthal ancestry on the X versus the autosome, we find it is substantially lower on the X versus the autosome, both in the autosome. There's a second prediction that comes out of this model, which is that hybrid sterility genes tend to be often found in genes that are highly expressed in testes. So we asked, what can we say about Neanderthal ancestry in genes that are specifically expressed in testes? And we find that both in European and East Asian, the Neanderthal ancestry proportion are substantially depleted in testes expressed genes compared to the rest of the genes. So the basic picture that's arising from this is there's been Neanderthal introgression, and on average, these introgressed alleles have been deleterious. However, there are specific regions of the genome where they could have been beneficial and resilient. So now let's move on to the second archaic introgression event in human history, and this has to do with the Denisovans. So this starts with a pinky finger bone that was identified in this cave in the Denisova cave in the Altai Mountains. And when the genome sequence was obtained from this pinky finger, it was found to be from a population that was neither modern human nor Neanderthals, but a population that was a cousin of Neanderthals. So it was a sister group. And like we did with the Neanderthals, we could ask whether there was interbreeding between Denisovans and other modern human populations. And what was found was that there was Denisovan gene flow into populations in present-day Oceania. So Oceanian populations today have, in addition to the Neanderthal ancestry, they have ancestry related to this group, the Denisovans. We can look at this more broadly. Here is a picture from about 120 populations that we surveyed for genome-wide portions of Neanderthal ancestry. And what we find is, again, there is a broad smattering of Neanderthal ancestry outside of Africa. Whereas when we look at the Denisovan proportions in these same populations, we find that there's essentially a peak of Denisovan ancestry in Oceanian populations. Not much outside, although there are small amounts of Neanderthal ancestry in populations in South and Southeast Asia. So current question, which many other groups are working on, is trying to understand the origins of these Denisovan ancestry proportions and to figure out what that allows us to infer about the Denisovan population. We can also build maps of Denisovan ancestry like we did for Neanderthal. So here is a comparison of Neanderthal and Denisovan maps, where we are looking at regions of the genome and tallying up which regions of the genome have considerable frequencies of Neanderthal and Denisovan. Like we saw for Neanderthals, we observe peaks of Denisovan ancestry. This is one example that's been very prominent. This overlaps a gene called EPAS1, and this gene has been found to be important for high altitude adaptation in the Tibetan population. And this introgressed variant is introgressed from Denisovans into this population. Likewise, we also see deserts, large regions of the genome that are depleted in archaic ancestry. For example, if you look at the desert on chromosome 7, overlapping FOXP2, it is a desert both for Neanderthal as well as for Denisovan. So some of these deserts are shared for both Neanderthal and Denisovan ancestry. And so again, this raises the question whether these are deserts generally for archaic introgression and what that tells us about the functional importance of these features. We can again do the same analysis where we tally up archaic ancestry frequency across the B statistic. And like we saw for Neanderthal ancestry, we find that Denisovan ancestry decreases as we move towards regions of low B statistic or larger functional extreme. And this is a trend we see both on autosomes as well as exosomes. We also see a similar pattern where on the X chromosome there is lower Neanderthal and Denisovan ancestry compared to the autosome. And finally, when we look at genes that are specifically expressed in testes, we find, again, there's a depletion of Neanderthal as well as Deficiency ancestry in genes that are testis specific. Yes.
I know the customs of the house and what is in the middle or in the end. Probably just again with the original video. Is this connected? Okay.
I can repeat the question.
Okay. So a couple of questions here. One is I think the evidence of FOXP2 has
been actually debunked, but I'm not an expert on it. I think Molly would know about that. But the other thing is, you represented this contrast between X and autosomes as kind of like a differentiating prediction between the incompatibilities versus purifying selection. But isn't it possible that you would expect the same thing under certain circumstances under the purifying selection Nielsen-Coop type idea? Because the effective population size and the X is lower, and depending on the distribution of selection effects and so on.
Yeah. So I don't know if I should. Hopefully people got the question. Do I need to repeat it? I guess the first question is about FOXP2. From what I understand, this is about the case for positive selection on the FOXP2 locus.
And its role in language evolution.
Yeah.
Like that. I think that was kind of pretty much debunked later on.
Yeah. Again, I'm not like strongly saying that FOXP2 is the language gene, and yes, there's plenty of evidence since that there are other loci with modifying effects. Yeah, so this is just like an interesting story, but there are many genes in that locus, and so I wouldn't like strongly say that the reason why there are deserts here is because of FOXP2 and its role in language or any of those statements. But coming back to the second question, which is about the X to autosome comparisons. Yes, so I think there are several aspects. One is kind of the genome-wide depletion. And also there is this trend that we see, which is this trend of B statistic on the X versus archaic ancestry. And that could also be a result of purifying selection under certain models. So again, having formal models of each of these is quite complicated because they are, we don't know the impact of selection coefficients on the X versus the autosome. Yeah, so I would say it's quite plausible that purifying selection is playing a role. The hybrid sterility model, yes, that's consistent with this, but again, you have these things about testes expressed genes that we also see, which is consistent with hybrid sterility. So yeah, I don't think there's kind of a strong preference for one model versus the other at the moment. Yeah, sex bias gene flow, we didn't model this, and that's insufficient at least to explain the magnitude of these effects. So it can explain some of it, but it's not something that explains all of the differences that we see. And again, I don't see exactly how that would also explain the B statistic correlation or these effects on testes expressed genes.
Isn't the B statistic calculated based on the models? Yeah, but I don't think that's a big issue. So when, for example, you generate like maps of B based on different human populations, including Africans, for example, you get essentially the same maps.
Okay, I mean, yeah, these are good questions. And it's good to have the B statistic thing, yeah, validated. Any other questions? I'll just pause here. Okay. Okay, so that's about the needs of an introgression. So now we move to the third type, which is introgression into African populations. So far, all of the introgression events that we've talked about have relied on the availability.
Without the microphone, sorry.
Okay, so everything we have said so far relies on the availability of these sequenced archaic genomes. So one question is the picture in Africa, which is less clear because we don't have access to these ancient genomes. It's been challenging to get ancient DNA at these timescales within Africa, although there is evidence from the fossil record about existence of these archaic forms. So the question is, what can we say about archaic introgression, if any, within Africa? So to look at this, we used two complementary approaches. So the first one is going to be based on something called the site frequency spectrum. So this is a measure that we compute, a statistic that we compute on whole genome data. And I'll tell you how we use this as evidence for archaic introgression. The second line of evidence is going to come from these introgressed segments that we talked about, but we are able to now estimate these introgressed segments without needing access to reference archaic genomes. So let's talk about the site frequency spectrum. So what is the site frequency spectrum? So we are looking at a population, and we're looking at polymorphisms in this population. Essentially, the site frequency spectrum is a histogram of the frequencies of these polymorphisms. So you would go to the first site and you'd count up the frequency of the site, or the allele at this site, which is 2 copies of A. You go to the second site and you'd count up that there are 2 copies of T. And so you'd build a histogram. And the reason why the site frequency spectrum is informative is that this histogram contains information about the demographic history of this population. In fact, under certain conditions, it's a sufficient statistic, so it has all the information that you can extract to be able to infer demographic history. So the idea is we can now try to build different demographic models to a site frequency spectrum computed from an African population, and including models that have archaic introgression involved. Now, the challenge with this approach is, in some sense, this site frequency spectrum is too rich. It has too much information. We'd have to have a detailed demographic model of the African population. And we are worried that we can't model every aspect of demographic history. So we're going to model a different quantity, which is called the conditional site frequency spectrum. So the CSFS. So for the conditional site frequency spectrum, in addition to our population of interest, the African population, we need an outgroup. So in this case, we're going to use Neanderthals or Denisovans as an outgroup. And we're going to identify those sites where we have the derived allele present in this outgroup. So we'll compare to an ancestral genome like the chimp and identify those sites where the outgroup, in this case Neanderthals, carry the derived allele. And we will only build the site frequency spectrum focused on this set of sites. So it's a very simple idea. And now the question is, why is this conditional site frequency spectrum a useful object? So this is a result that came out of this paper by Chen, Slatkin, and others, where what they showed was the following. So if you start with 2 populations, they split, and then they don't exchange genes. And if you ask, what is the conditional site frequency spectrum in the African population, for example, conditioned on Neanderthals carrying a derived allele, this CSFS looks uniform like this. And for this to hold, as long as these sites are evolving neutrally, and there is a few conditions like mutation drift equilibrium at the root, and there is no further contact between these 2 populations, this property of a uniform conditional site frequency spectrum holds. Now, why is this useful? This result holds independent of any population size changes in the African population. So this holds no matter what were the history of population sizes in Africa. So that's useful for us when we're trying to build demographic models where we don't have to worry about those kinds of complications. And as to why this uniformity assumption holds, the proof is actually quite straightforward and slick. Essentially, if you have mutation drift equilibrium happening at the root, this is what the site frequency spectrum looks like at the root. Conditioning on the derived allele, so the probability of identifying a derived allele here is simply given by the allele frequency. This is true because these are neutrally evolving, and this is a martingale process. And putting these two together, the conditional site frequency spectrum is the unconditioned site frequency spectrum times the conditioning, and so that leads to this uniformity. So this is theory. Now the question is, what does the CSFS look like in data? So if you look at 1,000 Genomes project, and here we are looking at the Yoruba population, this is the CSFS for the Yoruba population conditioned on either Neanderthal or Denisovan as the outgroup. So it's fairly clear it is quite far from uniform. Yeah.
And if you would use a chimp as the outgroup, would it be flat?
If you use chimp as an outgroup, you still have to identify a derived allele, right? So yes, as long as you're able to identify a derived allele, yes, it would be flat, or obviously a fewer sites, so it's noisier. This is also true if you look at other West African populations. So we looked at 4 West African populations from 1,000 genomes, and they all have similar looking site frequency spectra. There are possible explanations for this. So first one is technical issues. For example, errors, genotyping errors in the archaic genome, issues with identifying the ancestral allele correctly. So both of these could lead to deviations from uniformity. So we did some analyses. We found that the rate required to produce what we see here is too high. So that doesn't explain what we're seeing. Other possible processes that can produce deviations are things like GC-biased gene conversion, background selection, back and recurrent or recurrent mutations. And again, here we did a few different things. We filtered the data in different ways, looking at different classes of mutations, which are less likely to be affected by gene conversion, looking at different regions of the genome, which are less likely to be affected by background selection. And all of those give us consistent results in terms of the shape of this CSFS. So as we said, the CSFS is uniform, provided demographic history was Africans splitting from Neanderthals with no further contact. But we know that models of demographic history could be more complex. We know of events like gene flow into the ancestors of Neanderthals from the ancestors of modern humans. We know about gene flow between European and Neanderthal populations, and we know of recent mixing between Europeans and Africans. So the question is whether these kinds of deviations in demographic models could explain the CSFS. So we modeled what the CSFS would look like with these additional events. You do see some departure from uniformity, but still not enough to explain what we see. On the other hand, here is a model that does a much better job of explaining the CSFS. So this is a model where we have introgression into Africa after the split from Neanderthals. And this introgression is from a lineage or a population that split off prior to the Neanderthal modern human split. So we call these a ghost archaic introgression event. So it's a ghost event because we don't have a good candidate for this population. And of course, we need parameters to be able to fit this model. So once we fit this model with appropriate parameters, we find that we're able to recapture some of the features that we see in the data. We also explored other types of models, for example, additional Neanderthal-related introgression into the ancestors of Africans and Europeans. Again, it produces deviations from uniformity, but not quite what would explain our data and not quite to the same quantitative level as ghost archaic introgression. We looked at deep structure within Africa. So imagine you have 2 populations within Africa that are mixing genes over an extended period of time. Again, this also produces deviations from uniformity, but doesn't explain it quite as much as the ghost archaic introgression event. We can now fit parameters to this population, this ghost population. So we estimate that it diverged prior to the human Neanderthal split. We estimate that the introgression itself happened fairly recently into Africa. Of course, there could be an extended period of introgression. The proportion of introgression could be quite substantial, and these populations could have been very large. So these are all kind of statements that are likely to be refined as we get better samples and better models of demographic history. So in the second analysis, we went back to our question of whether we can identify these segments without access to archaic reference genomes. And the idea behind this approach is the following, where we simulate haplotypes under a specific demographic model. We then calculate population genetic features and train a model that predicts archaic ancestry without having reference to an archaic genome. The kinds of features we use are things that would be indicative of archaic ancestry. So one type of features will be looking at haplotypes at a given locus and trying to see if there is different populations of haplotypes based on the number of differences across them. So the idea is, if there is an archaic introgression, you might imagine that the pairwise differences across the archaic and the non-archaic group are going to be quite high compared to pairwise differences within a group. So seeing these kinds of features in the data can be indicative of archaic introgression. As a positive control, we applied this approach to Neanderthal introgression without using the Neanderthal genome. Turns out we can identify a fair percentage of Neanderthal ancestry, about a couple of percent using this approach. We also validated that we are finding similar patterns using this reference-free approach as we found with the reference-based approach. For example, we find the peak at this BNC2 locus. We are also able to reproduce these patterns of archaic frequency versus B statistic bin. So this is telling us in the case where we have a reference, the reference-free approach is being reasonable. And now we apply this to the African genomes. So applying this to the Yoruba and Mende, which are two of the West African genomes in the 1,000 Genomes Project, we estimate about 8% of this ancestry coming from this archaic ghost population. We can further analyze these putative archaic populations and compare them to existing genomes. For example, we can compare them to hunter-gatherer genomes, pygmy genomes, and known archaic genomes. And what we find is when we look at these archaic ancestries and compare them to the non-archaic, the archaic ancestries tend to be much more diverged compared to the non-archaic, including when we compare them to Neanderthals and Denisovans. So this would again be consistent with this phenomenon of a ghost archaic introgression. Okay, so in the final part, I'm going to talk quickly about some efforts to identify phenotypic consequences of introgressed Neanderthal DNA. And this project is bringing together what we know from evolutionary studies, see these maps of Neanderthal introgression, with these biobanks that are now pairing up genetic sequence with very diverse set of phenotypes. And the question is, can we leverage information in these large-scale data sets to be able to understand the phenotypic impact of introgressed mutations? And one reason why we need these large biobanks is because of the properties of these introgressed segments. So when we look at these mutations that are introgressed from Neanderthals, we call this NIMs, because these are essentially tagging these Neanderthal ancestry segments. We find that the allele frequencies of these mutations tends to be quite low. So these are fairly rare in the population. Imagine if there are 2% of the individuals having Neanderthal ancestry, the median allele frequency is going to be around 2%. So we need these large data sets to have any power to be able to identify meaningful associations between introgressed variants and phenotypes. So one way you can think of answering this question of whether there is some effect of introgressed regions and phenotype is by looking at this quantity called heritability. So heritability essentially tells us what proportion of variation in a phenotype is explained by genetics. And the way we do this is we fit what is called a variance components model, where the genetic effects capture the genetic variance component, the environmental effects capture the environmental variance component. And putting these two quantities together, we can estimate heritability associated with this phenotype. Now we can go beyond just understanding heritability from all genotype SNPs, and we can ask what is the heritability that comes from specific regions of the genome. For example, we can ask for those mutations that are inherited from Neanderthals, we can quantify the heritability from Neanderthals versus those regions that are non-introgressed and inherited from modern humans. So this allows us to contrast overall whether there's been an increased or reduced contribution of introgressed alleles compared to non-introgressed alleles. There are some technical challenges with doing this, and this has to do with the different population genetic properties of introgressed Neanderthal mutations with modern human mutations. As I said, they have lower allele frequencies in general than modern human mutations. It also turns out they have different LD patterns compared to modern human mutations. And so if you don't account for these additional population genetic factors, we find that comparing the heritability at these NIMs to non-NIMs will give us biased estimates. So this is a model which only fits the NIMs and nothing else. And on the other hand, if we fit models which account for both the allele frequencies and the LD pattern, we get estimates that are much better calibrated. So this is more kind of careful analysis that we have to do when we are analyzing these classes of mutations. So now we fit a model which has this component for Neanderthal mutations, these NIMs, along with these additional allele frequency and LD components. And now we can ask, what is the heritability explained by these NIMs for different complex traits? And how does it compare to non-introgressed mutations? So here we have a collection of complex traits across different categories. And what we are asking here is there any heritability, non-zero heritability explained by these NIMs for these different categories? And general picture is things like anthropometric traits, things related to blood biochemistry, things related to bone density, kidney-related function, and liver-related function. These are all from blood lab measurements in the UK Biobank. These tend to have non-zero heritability associated with these NIMs. Then we can ask, is there more or less heritability at these NIMs compared to non-introgressed alleles or modern human SNPs? And what we find is across all of these traits, it's either consistent with zero or there is a depletion in heritability across these different categories. So we think that this is, again, consistent with purifying selection acting against introgressed Neanderthal alleles, causing decrease in heritability at these introgressed SNPs. One question you can ask is this, we have already shown that Neanderthal introgressed alleles tend to be located away from regions under functional constraints as defined by the B statistic. So the question is this observation simply explained by the fact that these Neanderthal introgressed alleles lie away from regions of low B statistic or high functional constraint? So the nice thing about this model is now you can also fit models of heritability with the B statistic included as another component. Again, there are some technical challenges in fitting all of these components together. However, when we fit a model which has Neanderthal introgression with minor allele frequency and B statistics, so all of them jointly fitted, again, we find that at least for some of these traits, in trait categories, there is a reduction in heritability compared to non-introgressed alleles. So we don't think this is completely explainable by the fact that Neanderthal alleles are depleted in functional regions in general. So with this, I'll conclude. So what I've hopefully given you an overview of is the kinds of questions that we've been thinking about over the last several years in this realm of archaic admixture. And one of the key findings and takeaways for us is, in general, we do find archaic admixture when we have access to these reference genomes. However, as modern human genomes and the number of these modern human genomes increases, we are finding additional signals which were not possible or which don't require access to these reference genomes. So a question for us is, how pervasive are these signals of ghost introgression across different populations? And what might be the impact of these events on human biology in general? Another set of questions, which is more computational or statistical, is if we grant ourselves that this is kind of a picture of human evolutionary history, then we need to be inferring these models, which are no longer tree-like models of population relationships. So the question is, how do we actually infer these models, which are graph-based models or network models of human history? And this turns out to be a very challenging problem. We don't have good understanding of what methods work, what kinds of models can be inferred given the data that we have. So here's an example of a very early result that we have, which shows that even in the simplest cases, trying to infer these network or graph-based models can be very challenging. So this is a model which has essentially 5 populations, and there is one admixture event. And it turns out if you try to infer this model using popular approaches for estimating these network models, you make a mistake in where these admixture events are placed. So the question is, what leads to this kind of mistake? There are a couple of possibilities. One is this model is not identifiable. There are 2 models which give you the same score or likelihood. Turns out that's not the case. This is a case where you could have identified the correct model. But the problem is searching over the space of these graphs is challenging. So a lot of current models assume that these graphs are tree-like and try to make local moves to come to the correct tree. But doing that requires moving through some extremely low likelihood graphs to get to the correct graph. So as kind of a preliminary approach, we propose new moves in the space of graphs, which are able to traverse directly from the starting to the optimal graph. But this is, again, a very simple setup. We have no idea how any of these methods work in more complex and challenging settings. So I think this is a question which we'll be working on, at least for the next few years, trying to understand the possibility of inferring these kinds of relationships. With this, I'll stop by thanking all my collaborators over the years, and special acknowledgement to all the students and postdocs who led this work. And I'd be happy to take any questions.
What's to say that's the case rather than like multiple admixture? Would you actually say that those are the same one?
Yeah, we cannot say. I need to repeat it. Yes. So the question is, when we make inferences about ghost admixture events, what's to say that this is a single admixture event versus multiple admixture events from multiple lineages? Yeah, for sure, we cannot conclude that there was just a single admixture event based on this data. So our only effort in this kind of a modeling work, this kind of analysis, was to see if there was, whether the simplest models could be falsified, whether there was support against some of the simplest models, and to come up with a model which we thought was doing a reasonable job of explaining the data. But you're absolutely right. Yes, looking at some of these parameters, looking at the large effective population sizes associated with them, I think it's pretty likely that there were multiple populations. And yeah, so yeah, I think of these as kind of a starting point, and we need to go back and refine some of these models. Yes.
So following up on Hannah's question, so in principle, you could imagine, you know, maybe you do not have enough power, but you could see a signal that it differentiates the possibility of one versus several and so on, right? Because you could see heterogeneity in the divergence of the haplotypes and, you know, maybe the length of the haplotypes that you associate with the archaic admixture.
Right. Yes.
Yeah.
Yes. So the question is, yeah, maybe what approaches or what statistics might be informative of these kinds of events? Yeah, absolutely. Looking at diversity within the putative archaic population would be one way to look at this. There's another finding which I didn't talk about, and that has to do with timing. So did this admixture event happen before out of Africa? And so it's shared by African and out of African populations, or did it happen after? Or were there multiple events or a continuum, right? That's an interesting question. And if you ask me right now, based on what I've seen in the data, I think there's support for both pre and post out of Africa introgression events. And maybe it gets a little technical about why we think this way, but right now I think there's support for both. Again, that's hinting at complexity in these admixture histories. And so that's another way of using, for example, how much of it is shared out of Africa versus unique is another way of trying to get at these kinds of questions.
There's a question in the chat that you may want to.
Okay, let's see. So, okay. Are there earlier ones?
Yeah,
Is this okay? Okay, given the archaeological data suggesting high probability of admixture in the Levant, particularly in Israel, 100 to 115 BP, is there any genetic indication of introgression into the modern human genome? Or are all traces of that earlier admixture and AMH population lost? I see. So I think the question is these earlier introgression events. So there's been at least suggestions from the fossil record of the possibility of earlier admixture events in the Levant. And a question is whether that has had a genetic impact. Right? And so far, it's been very difficult based on the genetics to see if those have had any kind of impact. One way is if you were to get ancient genomes relevant to that period. So that would be one approach. I know several groups have tried and are actively working on this. So I think that would probably be the most direct way of trying to answer that question.